genome editing techniques with which we can modify the genetic composition. Okay, so first, such important genome editing technique that we have is nothing but CRISPR Cas9 technique. So this is one genome editing technique. Okay, so second technique is nothing but zinc finger nucleases. This is another genome editing technique that we have. Okay, then the next important technique that we have with us is Talon's technique. Okay, CRISPR-Cas9 technique is one thing, zinc finger nucleases is one thing, and Talon's technique is another thing. That means these are acronyms and these are the techniques. And along with this, so we also have another important genome editing technique called mega nuclease technique. Mega nuclease. Nuclease is nothing but basically it is nothing but an NGM. Okay, so NGM. So here, what is CRISPR-Cas9? Okay, so CRISPR-Cas9. CRISPR means, so when we talk from the perspective of CRISPR, CRISPR is an abbreviation for something called clustered randomly interspaced. Can anyone say? Randomly interspaced? Can anyone fill? Short palindromic repeats. Short palindromic repeats. So this is the full form of CRISPR. So you see CRISPR. So that is the abbreviation for that. Okay, clustered randomly interspaced short palindromic repeats. So here you have to understand what is this. Okay, what is this means? We have already said that genes or DNA will consist of genetic information, and this genetic information would be generally represented in the form of codons, would be represented in the form of certain codes. Okay, so like let's say AAG, or let's say TTC, or just I'm using hypothetically. Okay, like ATG. Likewise, so there would be three purines or three pyrimidines. They will combine and make a codon. Okay, likewise we have around 64 codons in entire genetic makeup. We have 64 codons, such codons. Okay, so these things would be arranged in this manner. Codons. Codons means that genetic information would be stored in the form of certain codes, that are called as codons. Like let's say codon would be, it will have like AGC or like GCC. This is a codon. Okay, likewise if we see in living organisms, so we have 64 such set of codons. Okay, so in human beings, so what will happen generally means such codons will repeat in genetic makeup. Okay, let's say for example GCC, GCC, GCC. Can you see this codon is getting repeated? Okay, such repetitions can be short, such repetitions can be medium, such repetitions can be long. Okay, short means basically 1 to 10, medium means around 10 to 100, and long means around from 100 to 1000. So that can be the frequency that how they can be repeated in the genetic composition. So such short repeats are known as palindromic repeat. Okay, one codon would be known as one palindrome. Okay, so that repetitions are nothing but palindromic repeats. Are you getting that? So here we'll be playing with these repetitions. So palindromic repeats means these things. So they will repeat like this. So here we will be altering such things. So CRISPR means, this is the full form, clustered, randomly interspaced, short palindromic repeats. Okay, and Cas9. What is Cas9 means? CRISPR associated protein. Associated protein 9. CAS, CAS means CRISPR associated protein 9. So that is the type of the name of the protein. Okay. So here in this technique, Cas9 acts as molecular scissor. We need a molecular scissor to cut. So there regularly we have said like restriction endonuclease will do that process. So here Cas9 is the molecular scissor that would be used generally in this technique. Okay. And mark that this technique is the most effective, it is the short, most effective and convenient method in the molecular biology that regularly scientists will use for genetic engineering. Okay, CRISPR-Cas9 technique. This is one important technique. Okay, so then talents. What do you mean by talents? Talents means it is also there is also a full form for this. So it is like transcription activator. Transcription activator like effector nucleases. Transcription activator like effector nucleases. That is the full form of talents technique. Okay, here also nuclease enzyme would be there, and that will be helping in the process of the molecular, that will be helping the process of cutting up the genes. Likewise, so here Cas9 protein. So Cas9 protein, so it helps in the process of that. So sometimes enzymes can also be proteins, proteins can also be enzymes. So this will help in the process. So these are the 4 important genome editing techniques that we have. Okay, so can you able to remember techniques now? Okay, so that is one thing. So CRISPR-Cas9 and talents, zinc finger nucleases and meganucleases, all of them are nothing but genome editing techniques. Can you able to remember? Yes, the process is same. In the place of restriction endonuclease, there this will come. That's it. Okay. In this technique, Cas9 will act as molecular scissors. In that, like nucleases will act like this. So that is same simple thing. Okay. So that is the basic techniques that we will use. Is that clear? So these things you have to remember.